Okay, so chapter 4, we're continuing this early embryonic development stage. And right now we're going to talk about the formation of the central nervous system, okay? Actually the central and peripheral nervous system. It's a process that we call neurulation. And neurulation is one of the first steps in an overall process that we call organogenesis. And really, this is the formation of the entire body plan, all the body structures. And it begins around week 3 and it ends around week 8 or 9. And as I said earlier, it is a highly vulnerable stage of development. So neurulation, as you can imagine, highly vulnerable because it's these weeks where you're forming your entire, for example, central nervous system. So the central and peripheral nervous system actually is derived from embryonic ectoderm, which is that top layer right there. So what happens in the process of neurulation is that a central portion of this ectoderm becomes what's called Neuro Ectoderm. Okay, so it differentiates into pre-nervous tissue. And there's this thing called the notochord, right? This little clump of cells right underneath it, underneath the ectoderm, that actually initiates this transformation. So the notochord is actually controlling this process of neurulation. So we start to form pre-neural tissue, and that pre-neural tissue is starting to form something called a neural groove. Okay? So it starts to invaginate. And we can see over here on the left, this is an 18-day embryo, and we can see the invagination on the top. Okay, so 17 days, we have no invagination, just a primitive streak here. And then over here, we're starting to see folding. So from there, that neural groove starts to get more and more invaginated, and it starts to actually become what's called a neural fold. And around the time where we form the neural fold, there are these cells that migrate out of the neural fold and differentiate into another cell type. There's these clumps over here, and that cell type is called the neural crest. And the neural crest is going to become our peripheral nervous system. So this neural fold is our central nervous system, and the neural crest is our peripheral nervous system. Now, so you can see, so it's kind of hard to imagine because it's so amazing, but this neural fold, so this thing that looks like not much of anything is already got a polarity to it. So this embryo that really just looks like this thing, this region is already designated as the head region, and this region is already designated as the tail region. So the neural fold tissue up here is already destined to become the brain, and then down here, it's destined to become the spinal cord. So this neural crest that's forming alongside of it, right? At the upper regions of the embryo, it's going to become the cranial nerves, and at the lower regions, it's going to become all of the spinal nerves. Okay? And you see these red bulbous portions of mesoderm, okay? And they are going to form what's called the somites. So these bulbous regions of mesoderm that are on either side of the neural fold are going to become the somites, and they are going to form all of the segmentally innervated muscles of the trunk of the body and the vertebral bones. Okay? So you can see how the spinal nerves and the cranial nerves are all forming alongside some of the targets of innervation, and also the bones that are going to encapsulate this nervous tissue. Okay. So this neural fold gets deeper and deeper until finally it forms what's called the neural tube. And the neural tube, this neural fold starts to seal off to form the neural tube around the center of the embryo, and then it seals in both directions, right, towards the head and towards the tail at the same time. So it's like a two-way zipper. It zips up and it zips down. Okay? And the closure of the neural tube forms eventually the brain at the top, okay, and the spinal cord at the bottom. And you can see these somites are becoming much more clear. They look like these little bulbous ridges on either side of the center of the embryo. And by around 20 to 30 days, or around 4 weeks, these somites become, 4 weeks embryonic age, these somites become very clear. Okay. So this is just another view of this process. So 18 days, we're just starting to see the neural fold, right? And then 20 days, we start to see the closure of the tube in the middle, formation of the somites. 22 days, this tube is starting to close more readily, and it's got more somites forming, especially towards the top of this embryo. And then by 23 days, the neural tube, the vast majority of the neural tube has closed. It only remains slightly open at the bottom, at the bottom and at the top. Okay? And that we call the Neuro Pores. So there's an anterior and posterior Neuro Pores. And eventually these Neuro Pores are also going to seal, but they're going to seal once they form at the top here, once they form the cerebral hemispheres and the rest of the brain, and towards the bottom here once it forms the fully forms the spinal cord. Okay. Now, the wide variety of congenital malformations that we call neural tube defects really starts here in neurulation, and it's caused by a failure of the neural tube to fully seal, to fully close, and sometimes you have problems at the rostral end or the head end. Okay, so you can have portions of the neural tube not closing at the top. You can have portions of the neural tube not closing at the bottom. The ones at the bottom are the causes of your spina bifida cystica and the other forms of spina bifida. And the other closely related disorders to the neural tube dysfunctions or neural tube disorders has to do with the malformation of the somite tissue. And that's where you start to see issues of abnormalities in vertebral bone formation and fusion. And at the end of this here, just going to close with a pretty picture. So you see this, this is actually very early neurulation where the tube is just starting to seal. We don't even really have any somites at this point. And it's an actual photo of an embryo forming its nervous system. Really amazing.